Hello everyone. So today I'm going to be guiding you through how to use our Autotrace tool within our Phoretix Toolbox image analysis software to automatically pick out things like cells or different objects within an image so that we can then measure their volume, measure their area, get different quantitative measurements from that image. So you can see here, I've got a classic example of something that you might want to measure, some cells down a microscope, and you might want to see over time if your cell's getting bigger or smaller, you might have a drug treatment, that kind of thing. So this obviously all starts with measurements. Now, I'm lucky here in this example image that I've grabbed in that it contains calibrated information. So the metadata within the TIFF file for this image, you can see that it actually has representative information in millimeters. It means that this image is calibrated and we've got that information to go along with the image. Now, if we didn't have this information, we would still be able to measure objects in pixels, area in pixels, that kind of thing. But because we have got the scale information baked into the image from the microscope that this was taken on, we can represent everything in millimeters, which is a bit nicer. We don't need to do any calibration or anything like that. So this is a high quality image, 16 bits per pixel, TIFF file format, so we should get some pretty good results when we're trying to define boundaries and automatically trace around cells. So I'm going to come through to the Analyze tab and I'm going to zoom in. So I'm just using my mouse wheel here to zoom in and holding shift for the shortcut there. I'm going to grab the Autotrace tool here. I'm going to place kind of the center of where I want to do my auto trace, and you can see here it's made an attempt. What we do here for the auto tracing is we basically set a threshold pixel value, and that defines whether everything below or above that is selected. So it works really well on something with a bright white or a bright or a dark black background. To change the settings, again, I can use the settings here on the left hand side, so I can click and drag these to change the tolerance level, or I can use my mouse wheel. So you can see the mouse wheel allows much finer control when I'm changing these different parameters, when I'm working out exactly where I want to place the edge of my threshold. And when I'm happy with kind of where the edge of my threshold lies, I can click on complete shape, and then that gives me the shape. So instead of me having to trace around kind of the edge of my boundary here for my cell, I can just click on OK, it's grabbed the edge of it, and like I said before, because we've got millimeter information here for this example, we can get different areas, heights, widths, example, things like that. So we can get different measurements, we can start doing area measurements and things like that, getting volume measurements, and of course, and because this is built into our Phoretix suite of image analysis software, of course, all of these different measurements can be exported, can be copied and pasted to Excel. You know, you can do further calculations or measurements with these, or have a place to store them if you want to do things like changes over time, that kind of thing. So I know the threshold that I set for this cell, and then I can start applying that to other cells. So if I come along and grab this one, you can see it's remembered the tolerance level that I used before. I can complete that shape, and I can compare them on a like-for-like basis. So I can make sure that the settings that I use to capture this cell are used for the other cells as well. This is very useful and very powerful because it allows you to keep a constant kind of detection setting between all of your different cells, rather than if you're using something like ImageJ or some kind of, MS Paint or Photoshop or something like that. You're going to run into operator bias, inter-operator bias, and intra-operator bias. So two different people might place the edges of a shape in different areas. Some might crop closer to the edge, so some might come here, some might come on the outside of the edge. So when you're trying to compare measurements between different working groups, different people, there's no kind of level of standardization. It's very personal. So that's one of the main sources of variation when it comes to image analysis, and especially in terms of microscopy. By having a standard setting that you can apply for everything, you kind of normalize your results, you kind of standardize your results across different operators, which gives you much more uniform reporting, much more reliable results. So by using the Autotrace tool, we can get rigorous, reliable results for things like cells, wound assays, if you select the center of the wound, for example, things like that. And of course, all of these measurements you can export and you can do different manipulations with further down the line. If you'd like to try a free copy of the Phoretix Toolbox software, please check out the links in the description below. And if there's anything else you'd want to see included in the Phoretix Toolbox software, please try the software, leave us a message, contact us through the website, or drop a comment on the